Abbiamo una sessione con 4 interventi di 20 minuti l'uno. Inizia Dr. Paul Thomas, University of Adelaide, Abnormal Cell Sorting, Underlies the Unique X-linked Inheritance in PCDH19 Epilepsy.
It's on? Yeah, perfect. Thank you. Well, thank you, Francesca, for the opportunity to speak here today. It's a real pleasure, and also great to see some colleagues who in the field that I haven't met previously. That's been also a very good experience. I'm going to stand up and talk. I think if I sit down and talk, I might fall asleep. So in order to stay awake, I'll be standing for the entire talk. And today I'm going to tell you about something which is a little bit different to what we've heard about already. I'm going to tell you about our work to try and understand what's happening actually within the brain of children with PCDH19 epilepsy, but using cell-based models and mouse models as well. I won't really go through the background very much. We've heard all this already. The only point I'd like to make is that it's such a highly variable phenotype in a clinical sense, and that's something I want to come back to later. And we've also seen already the inheritance of this, very unusual inheritance of the disorder, whereby females are affected with 2 X chromosomes, one of which contains the mutation, whereas carrier males are not. Very unusual. And we haven't heard so much about the protein itself or the gene, so I'll introduce that now as part of the talk. This is from a review from Depien and Legurn, both in the audience today, describing really there's very many mutations that cause this particular form of epilepsy. The gene itself is quite a large gene, and most of the mutations cluster here in exon 1, which in the sense of the protein corresponds to the part of the protein that sits on the outside of the cell. So let's talk a little bit more about the protein itself. It's part of a cadherin family. Many of this you will know this already, but I'll just use this as way of introduction for those people who are not so familiar with the protein. It's a form of a protein which basically has 2 components. There's an outside of the cell component and an inside component. And basically it's from a large group of proteins which helps cells to stick together in a very specific way. There are many proteins of this nature or of this family within the genome. And PCDH19 is just one of many proteins with this particular structure and function. So we've been trying to understand how this protein works, and we've initially used cell-based models because they're much easier to do experiments and gain information about the impact of mutations on the function of the protein. So this is just a little bit of data from our lab that's consistent with other reports in the literature, and it just basically tells you that PCDH19 is a so-called homotypic cell-cell adhesion molecule. So what we've got here, and I'll show you a few examples of this as the talk continues, 2 groups of cells in different colours, and in this case, both of the cell groups express PCDH19. And when you do put them together and allow them to mix within the dish, they form these very heterogeneous aggregates. And this tells us that PCDH19 does indeed stick cells together. But when you do, as does PCDH17 and PCDH10, because they're all part of the same family, even though they're slightly different proteins. But if you now mix together PCDH19 expressing cells, shown here in purple, with PCDH17 cells, shown here in green, they form separate groups, so they segregate away. They both have cell-cell adhesion activity, but they can't stick to each other, so they form separate groups of cells. Now this slide we've also seen already, and this is one that I think I've shown many times in seminars over the years. And it's basically this cellular interference model, which was proposed by Depien et al. A number of years ago, which is an attempt to explain this very unusual inheritance of PCDH epilepsy. And really, as we've seen already, basically the idea is that in affected females and also mosaic males, there are 2 populations of cells. And these 2 populations of cells either have a functional PCDH19 gene or a mutant, non-functional PCDH19 gene. And because of this, they form separate interactions or networks within the developing brain, and this is the basic mechanism that gives rise to the phenotype. And And the question really has remained unresolved in the literature, despite this being a very nice theory of the mechanism of this particular disorder. Is it correct or not? So what kind of experiments can we do to test it? And there are basically two things I want to tell you about today, or the two questions that we started with this project. How does mosaic expression of protocoherin 19 lead to epilepsy at the molecular and cellular level? So what's the mechanism? And why is it that complete loss of protocoherin 19 does not cause epilepsy? So why is it that males who carry the mutation in all of their cells, why does that not cause epilepsy? And part of the understanding that we've come to, and the mechanism that I'll propose, has come from looking at other protocoherin molecules within the genome and recent studies looking at their function. And this is quite a complex looking slide, but basically the idea is relatively simple. And this is the essential idea, is that protocoherins can form combinatorial adhesion codes. So what I mean by that is that if you have two groups of cells, each of which, and you've seen this before, each of which expresses multiple protocoherin molecules from different genes, if they're an identical combination, the cells will mix together. However, if you just alter that combination by just a single protocoherin, that will cause the cells to form these clusters that are segregated away from each other. And there's quite a lot of evidence in this paper which they use to build up these models, and basically the idea is that there are very specific interactions that occur within the protocoherin molecules that are at this interface or at this interface, and they're called cis or trans interactions, which enable these lattice-like structures to develop at adjacent cells, which are very large if there's an identical combination, but they're very small if the combination is not identical and it's not sufficient to keep the cells adhering to each other. Okay, so the question we had then was, from this related group of protocoherins, this clustered protocoherin protein group, does protocoherin 19 have these same features, functional features? So we went to explore this. And in order to explore this model, we need to answer a number of questions. Do protocoherins, protocoherin 19 for example, have overlapping expression with other family members within the developing brain? Do they form specific trans interactions? And does it form cis interactions as well? And to cut a long story short, the answer is yes to all of these questions. So here is a bunch of so-called in situ hybridization experiments. So this is expression data telling you that the protocoherin belongs to this family of proteins that are active and have overlapping expression throughout the brain development in mice, and it's also the same in humans, we believe. This tells you that there are specific trans interactions from this data that I've been showing you already, and we've naturally now looked at the proteins in much more detail and looked to see whether they have this ability to interact with each other in cis, and that is also the case as shown by IP analysis. So in other words, this clustered group of protocoherin molecules and the non-clustered protocoherin molecules seem to have the same kinds of properties. And that's enabled us to study the protocoherin epilepsy at a cellular level just by expressing different combinations of protocoherin molecules within cells within the dish. So this is the idea. So the protocoherin, non-clustered protocoherins work in a combinatorial fashion to generate unique adhesion specificity. So here we go. So here we have two populations of cells, but now instead of just expressing a single protocoherin, we've got them to express multiple protocoherins. In this case, protocoherin 19 and 10 in both populations of cells, and you can see that they mix together quite readily, as you'd expect. But again, if we just alter a single protocoherin, so for example, if we keep the green population the same throughout these experiments, but if we delete protocoherin 19 from one population, we find that they no longer mix together to give you a heterogeneous population, but instead they start to segregate away from each other and form separate groups or separate clusters within these larger aggregates. If we substitute one of the protocoherins, this time we put in 17 instead of 10, we again see separation of the clusters and segregation. And the same thing again if we add an extra protocoherence. So now we've got 3 versus 2, we see this segregation phenotype. This is all in cells within the dish. So that's not a bad start. It's giving us some information about the way protocoherin 19 interacts with other proteins. So then we started to look at mutations that cause this form of epilepsy and ask the question, how does that affect the cell's ability to form interactions with other cells in the dish? So here we've got wild-type protocoherin 19, again showing that it can form aggregates within the dish. And here are 2 mutations that cause this form of epilepsy. One of them we've heard of already this morning. We had a very nice description of the variability of the clinical phenotypes associated with this particular one. And here's another relatively common, as I understand, mutation as well. And the point that I'd like to make is that these have absolutely no activity within this assay. So they seem to be functionally complete loss of function. This is just some data to show you that the proteins are indeed expressed within the cells, even though they have no functional no function within this assay. So that means we can essentially model protocoherin 19 epilepsy, and I've used this in the context of the original cellular interference model. So here's our idea so far. Here's a normal individual with multiple protocoherin molecules that are identical on each of the 2 cell types, and that interacts very nicely. Hemizygous males have a loss of function in the protocoherin 19 molecule. So here we can put that into the assay as well, and we ask now what happens between cells that express mutant protocoherin 19 in 10 on one and mutant protocoherin 19 at 10. And again, these form very nice interactions, telling us that the hemizygous males, this is an explanation for why the hemizygous males are not affected, because these essentially are the same as these. And lastly, the experiment where we put in a mutant form of protocoherin 19 with 10 and the normal protocoherin 19 with 10, and again we see this segregation phenotype. So the bottom line here is that there's a combinatorial aspect to the way these protocoherin molecules work within the cell to bring cells together during development. So we were very interested not only to study this in vitro, that is just in cells within a dish, but also to study it in vivo, because really that's what we're really driving towards. That's what we want to understand. What happens in brain development when you lose a copy of protocoherin 19? And this was technically very challenging, and I won't go through the details of this, but we managed to create an animal, a mouse, in which we could identify the protocoherin that's being expressed from that particular chromosome, and this is a wild type form. So a normal form of protocoherin 19, but we put a little tag on it just so we could trace its expression in the brain. And that worked very well. This is just data to show that it worked at the protein level, both using so-called Western blots as well as immunohistochemistry. So we managed to tag the protein, the wild type protein. And a little bit about where it's expressed. Well, basically it's expressed in both of the key neuronal cell types that are created during brain development. There's so-called excitatory neurons that are generated in the cortex. They express protocoherin 19, as do migrating interneurons during their development as well. For those of you who are sort of into cortical development in a sort of more detailed way, we find that layer 5A seems to be the site at which protocoherin 19 ultimately becomes restricted to after brain and cortical development is complete. But we're still actually looking in more detail into the cells, the exact cell types that express protocoherin 19. This is to show you that the interaction between protocoherin 19 in vitro that I've showed you already also seems to be occurring in vivo. So this is again an IP analysis from Rahman, who works with Joseph, who has now evidence for interaction between protocoherin 19 and another member of the family, protocoherin 10, in the developing mouse brain. So we think we can translate reasonably well from cells within the dish to the developing mouse brain. So to complete the mouse development work, we needed to have a reporter gene, or in other words, a way of tracing the mutant form of protocoherin 19 as well. And it turns out we'd already published a mouse that has this particular reporter in place. So this is a beta-galactosidase reporter gene. And this blue staining you can see here corresponds to the location of cells which express the mutant protein within the developing brain. So this gave us all the tools that we needed to do the key experiment, and this is the critical experiment for the talk. Basically, what we wanted to do was to create mouse brains that had the same genotype as what we see in the girls with PCDH19 epilepsy. So to do that, we needed to bring together I'll keep this in green for the rest of the talk. This is the wild type protein, and this is the mutant protein. Or in other words, this is the wild type allele, this is the mutant allele, and ask the question, do these cells segregate away from each other during brain development? So here's the control first. So this is just an animal, it's a female animal that has essentially a wild type genotype, and what we've got is this tagged molecule showing the location of cells which express this HA flag, and you can see that this is the pattern that we see when the brain is wild type. Now, if we do that cross that I mentioned earlier and bring together the mutant cells and the wild type cells within the same brain, this is the pattern that we see. Quite a striking difference in which we can now see the developing cortex here and also the hippocampus. The cells that are expressing the wild type PCDH19 segregate away from those that are mutant. And I can show you now the location of the mutant cells as well. This is the same brain now expressing the X-scale stain shown here in purple. And if you put those two together, what you get is a nice pattern, a complete picture of the developing brain in which we can see the wild type cells in green and the mutant cells in purple. So there's quite a clear segregation of the two cells. And this is in complete accord with the original cell segregation phenotype that Christelle proposed a number of years ago. Okay, so I can tell you a little bit more about the phenotype and what we've learned about it over the probably last, year or so that we've been studying it, a bit more than that, 18 months. This is just some quantitative data to show you that the effect is real. I think it's quite an obvious phenotype in any case, but the reviewers of our paper that we submitted made us do the quantitation study, so that's why I'm showing you that here. It took quite a bit of work to sort that out. And this is just more data showing you that the mutant cells and the wild type cells are quite clearly segregated. And this occurs in a number of parts of the brain, as I said. It occurs across the cortex here, and it also occurs within the developing interneurons as well. So this is that region down here, the median ganglionic eminence. You can see these stripes of cells that appear where again we see segregation of the two different types of cells. So when does it occur during brain development? That's a question I think that's quite interesting. So to answer the question of when, we had to go back and check when is the first time that we see protocoherine expressed during mouse brain development. So mouse brain development really doesn't, it gets the very earliest part of the embryo, which is called the brain, is around 7.5 days, the neural plate, and then the primitive brain gets formed at around 8.5, and at about 9.5 days of development, so this is very, very early, we start to see PCDH19 expressed, and that's shown here on the left-hand side. And this is in the cells that will ultimately form the cortex. So 9.5 days is the earliest time point at which we see protocoherine expression. And now I'm going to show you what happens during that kind of time point to the brains that are a mimic, that are the same genotype that we see with the girls with PCDH19 epilepsy. And that's shown here. So this is 9.5 days of development. This is the control, and this is the affected genotype here. And you can see even by 10.5 days of development, so really there's only been expression of PCDH19 for around a day within the developing brain, we can already see this nice segregation phenotype occurring. So in other words, the rearrangement within the brain, the segregation occurs at the very start of brain development. This is to tell you, for those of you who are interested in what happens to the brain after embryogenesis. We see the segregation phenotype is maintained after birth, so this is at P7, so 7 days after birth, and actually it continues on to at least day 42 after birth. We haven't looked later than that. This is the point that I wanted to come back to in terms of the variability. So as we've been hearing all day, it's such a variable syndrome in terms of the clinical phenotypes associated with what could be considered the same kind of genotype, really a loss of function on one allele and wild type function on the other allele. And the point of this slide is just to show you that when we look across many different examples of the same affected female genotype, each one is unique, each one is different. And we think this is because the exon activation pattern that occurs in the brain is going to be unique to each individual. And because that then causes some cells at the very earliest stages of brain development to either express protein adherent 19 or not, it means that the pattern that we see in terms of this segregation event is also unique and different in each of the cases that we look at. So I think this provides some insight into the phenotypic variability that we see in patients. This is a rather complicated slide again, but this really comes down to one thing, and that is that the ability of two cells to come together when they express the same range of protein adherent 19 molecules is very high. Whereas if you make a change to just one protein adherent component, for example, protein adherent 19, the chance of those two cells coming together and having a stable interaction is much, much lower. At least that's what the modelings tell us, and it's completely consistent with the data that we see in vitro and in vivo. And that's just shown here. So we wanted to test this a little bit further and do just one more experiment that I'll tell you about, and that is to ask the question, could we rescue the phenotype? Could we, by altering, by changing the mutant such that now all of the cells carry the mutation? So here's our starting, here's the experiment. We start off with this genotype. We've got a wild type allele and a mutant allele. If we alter the wild type allele so that it no longer has a functional protein adherent 19 gene within it, and we change it so that it's now non-functional, do we rescue the phenotype? In other words, do we find that these mutant cells now do not segregate away from each other, but rather form a normal pattern within the brain? So we did that experiment, and we did it in a quite a technically challenging way, which again I can explain if anyone's interested. But I'll just show you the result of the experiment. So here's the control, and this is the segregation pattern that you've already seen. But if we change this now so that this wild type allele has now been knocked out, so it's no longer functional, what we get back within the brain is, instead of having the segregation pattern, we now have a normal pattern within the brain again. So it's returned back to its normal pattern. So the last thing I'll tell you is really what we're not only interested in the way the cells interacts within the brain, but also can we find evidence of this segregation phenotype giving rise to altered neuronal activity within the brain. So this is just one short experiment that we've done in collaboration with Kay Richards and Steve Petru in Melbourne, and the results were quite striking. Here's the ECOG trace of a wild type animal. This is the ECOG trace of one of the heterozygous animals, so this is a mimic of protein adherent 19 epilepsy. And here is a hemizygous, actually it's a null individual, so it's a female as well, but there's no functional protein adherent 19. And again, we see this difference between heterozygous and complete knockout in terms of the ECOG phenotype. And we looked at this in a little bit more detail with Kay and Steve's help. And it turns out that the number and duration of the spike wave discharges within the females that have the heterozygous genotype is significantly different to the hemizygous males as well as wild types, as you would expect from this particular unusual mode of inheritance. And the last thing that we've done in collaboration with Simone Mandelstam in Melbourne, and also Ingrid Scheffer to some degree, has been helping with this. We also find some examples of, through MRI, developmental abnormalities within the brain. They're extremely variable, unusual areas of sulcation, focal cortical dysplasias. None of the patients are the same in terms of what we see. We don't really know the significance of this, but I mentioned this for this particular audience as I thought it would be of interest, especially since the clinical session that we've just had, which mentioned the other MRI paper that's recently been published. So in summary, this is the last slide. So in summary, this is this is our functional model of how we think this particular unusual form of epilepsy occurs within the developing brain. Essentially, we see that populations that either express the protein adherent 19 molecule, or those that don't, form separate clusters. They segregate away from each other during the very earliest phases of mouse development. And we have a fairly good handle now on the molecular details that causes this cellular segregation. And what's more, ultimately, this seems to, definitely does correlate with altered brain activity, such that the animals that have this unusual segregation of early neuronal precursors has a kind of eleptiform brain activity, as I showed you a moment ago. And we think this provides a cellular mechanism for predicate hearing 19 epilepsy that's been, I guess, proposed a number of years ago, but now we put some flesh on the bones of that particular cellular interference model. And our challenge now is really to connect what's happening during brain development with the unusual brain patterns that we see, to try and understand how it is that this cellular phenotype ultimately gives rise to the abnormal circuitry that occurs within this particular mouse model. So on that note, I just want to thank particularly Dan Pedrick, a fantastic PhD student who did most of the work that I've shown you here today. He's now finished in the lab. He's gone to Stanford for a postdoc. He was ably assisted by many within my group. I'd certainly like to thank Joseph for a long-term collaboration. It's been terrific. And also Raman, who did the IP, and our collaborators and funders, and thank you for your attention.
So thank you very much for a great talk and extraordinary data. I suggest we have just a couple of burning questions and leave the rest of the discussion at the end of the session, because this is a basic science session, so questions may be interconnected. Domande? Prego. This is a microphone missing. There. For you.
Hi, thank you. That was a great talk. I have a question regarding of when you eliminate protocadherin 19 from your protocadherin 19 tagged animals, do you do it in a way where it's eliminated from the beginning, or are you doing it acutely?
Could you just repeat the very end of the question? Do we do it in a way?
Do you do it in a way where it's off from the beginning of development, or do you do it acutely?
Yes, we did. So we injected zygotes, so one-cell embryos, to remove it at the very start of embryogenesis. Yep. It was done using CRISPR, so we just cut out the exon 1. Yep.
Beautiful talk, Paul. So in your cell aggregation assay, did you ever compare just overexpressing double the amount of PCDH19 with your regular overexpression to see if you get the same phenotype?
No, we didn't do that experiment very carefully. They're all overexpression experiments, but we haven't carefully measured the level of protocadherin in different cells or tried to do like twice the amount of plasmid in one versus the other. We recently, again, because we were asked to check the level of the different protocadherins by Western blot, because they're all tagged, and there's not a lot of actually variation between them at a global level, but we haven't actually done the experiment that you're suggesting.
Last question, yeah.
Thank you, Thomas, for your very beautiful speech. I was surprised by the distribution of in cortical column. So we have to focus on the interaction between the migrating cells and the guiding cells. Do you think so?
Yeah, so initially the protocadherin molecule appears to be expressed in all of the stem progenitor cells before differentiation starts. Once differentiation starts, it is down-regulated and seems to become, remain in the stem progenitor cells for a period of time, but it seems to be down-regulated during differentiation. So the question you're asking about how the columns, I guess, are affected by the loss of protocadherin 19, there's probably a loss of the protein at the same time that the guiding cells are being used as a scaffold for the next differentiation stage that comes through. As you know, it's a very complex mechanism, and we haven't looked very carefully at each stage to see exactly which cells still express protocaterin 19 and which ones don't. But clearly that segregation is occurring even before there's overt differentiation. So by 10.5, we already see segregation occurring. So the differentiation is occurring in the context of an already segregated cortical plate.
Yeah, but what is your explanation why there is a cluster segregation and that segregation doesn't seem to be just random? What is your explanation for the cluster segregation, which you have demonstrated? Yeah.
So at the same time the cells are segregating from each other, they're also dividing. So what might initially be a relatively small group of cells that have become segregated from their neighbours, they will continue to divide and also continue to segregate is what I believe is happening. Yeah. So the clones themselves become larger.
Great, thank you very much. Next speaker? Who's next speaker? Okay. Silvia Bassani, National Council of Research, Milano. The dual role of the female epilepsy protein PCDH19 at the synaptic and nucleus. There is some typos in this. Yeah, okay, anyway. Va bene, 20 minutes?